Hey, biotechnology. We're going to continue learning about antibodies and the immune system, and what we're going to talk about today is probably very useful as far as what's going on in testing and whatnot. So, we're going to talk today about immunoassays, right? So, well, I don't have to do that. So, immunoassays, right? So, what they do is they take advantage. Of antibodies. So remember, if we have an antibody, it's a different color, right? So antibodies, remember, are specific for epitopes. So these are antigens. So an antigen is anything that's foreign to the body. Like if you have allergies, it may be in the springtime when flowers are blooming and the pollens get into your nasal tract and it causes you to get stuffy noses. Well, those little pollen grains are what they call antigens, and antigens are things that are foreign to your body. And remember that the antibodies that you produce look for epitopes, and those epitopes are little proteins on the surface of the antigens. Right? So antibodies are very specific for the antigens they look for. So we're able to use that type of a system to detect things that we want to detect. So we're going to go over this a little bit. So, and I'm going to come back up to this slide at the very end because it's really important to talk about. So they take advantage of the specificity. So the specificity right here of antibodies. Specificity means it only recognizes one thing, right? So we use it in qualitative tests, right? Qualitative tests is, do you have it or do you not have it? Test kits like HIV, pregnancy test, and then we'll talk about the COVID test, right? The antibody test is for that. They have GMO test kits. So the one we're going to concentrate on today is called Elisa. So we're going to focus today on Elisa. We'll talk a little more later about octolony and Western blots, but today we're going to focus on Elisas mostly. So let's get going. So Elisa stands for enzyme linked. So if you look right here, enzyme linked immunoabsorbent assay. It's a very common test. It's very useful for detecting proteins. So we use it quite often. We were going to do a really cool lab with Elisa, but things happen. So I'll figure something out for that. But types of Elisa. So make sure that you're able. I'm going to expand this a little bit, I think. All right. So the different types of Elisa, you'll have to be able to pick up a lineup, kind of. So we'll talk about direct Elisa first. So I guess before we talk about Elisa, I'm going to show you a picture and then I'll come back. So this right here is a microplate. But a microplate, if you remember when we did, we made pictures using dyes, right, in our pipette lab. So this is a 96 well plate, and it has a plastic on it. The plastic will bind proteins. So we're talking about Elisa, right? So a direct Elisa. So antigens, remember antigens are foreign particles, right, are immobilized, right? So what happens is, if I have my well plate here, so my well plate here, the antigen sticks to the bottom of the well plate, right? And then the primary antibody. So I have antibodies that have been produced. Maybe I injected a rabbit with the viral antigen, and the rabbit produced antibodies. And on the antibodies, this is, I'll use red there. This is an enzyme. So it's an enzyme that produces color. So what happens is, if I have that viral antigen, if it attaches to the bottom of my well, then I have an antibody, right? And that antibody, I have put an enzyme on, right? It's enzyme linked, so I've linked an enzyme to that antibody. So if if my antibody attaches to it, then I can hit it with a buffer, and that buffer is going to produce a color. And that color is going to tell me if the patient has that antigen or not. So that's called direct, and it's called direct because the antigen, right, is read directly from the antibody. Right, so all right, I'll talk to you about that. So we have indirect or direct and indirect. So if you look at it, it's pretty similar. So an indirect, again, I have the well, right? My antigen attaches to the well, and now I get a primary antibody. So this right here would be my primary antibody. Right? So my primary antibody attaches to it, and then I have a secondary antibody. Right? And that secondary antibody, just like here, has an enzyme that produces color. So the question is, why do we do indirect instead of just doing direct? Because they're very similar. Well, the thing is if I do indirect, I can get multiple secondary antibodies that attach to this primary antibody, and I get a greater response. So the pros, right? The indirect is more specific. So I get fewer false positives, right? And fewer false negatives. Okay, so it's more specific. It's a little more costly, right? But the product is much better. So indirect would be used much more than direct, right? All right. So labeling, this right here is, right, color, right? So it's just an oxidation reaction, emitting a color. Okay, so that's all we mean by labeling. And I'm not going to go much more into that, right? So the epitope is not labeled. In direct, the primary antibody is labeled. In indirect, the secondary antibody is labeled. All right, so if you look at, here's another example, just a little diagram of indirect for you, right? So the first thing I would do in indirect Elisa is I would add the antigen to the well. When the antigen sticks there, I would add a primary antibody, right? Then I would add a secondary antibody. So this is my primary, and this is my secondary. And then I would add a buffer, right? So I'm adding TMB, which is a common one we use, right? And this is an HRP, secondary antibody. So the TMB then acts with the enzymes to produce the color blue, right? So my wells light up blue. So if you look at the wells here, right? If it has a darker blue, it's a greater positive. A lighter blue is a lesser positive. So it gives you kind of that colorimetric quality to it. All right, and that's what we were going to do. We were going to do indirect Elisa. All right, so indirect Elisa is good, but we also have what's called sandwich Elisa. Right, so this is probably the most common one used to detect diseases and stuff like that. All right, so let's look at what it has. So things that make it different. All right, so if you look at it over here, I have a capture antibody. So now my antigen is not attaching to the plate. My antigen is only attaching to a capture antibody. So the reason why this makes it better is if I just had a well and I put blood in the well, I'm going to get this protein attached, I'm going to get that protein attached, I'm going to get that protein attached, I'm going to get that protein attached, right? So all different types of proteins are going to attach. And then I'm going to hope that my antibody is going to attach to the one I want it to attach to. So it's not as specific. All right, but when I have sandwich, right, now I have a capture antibody. So the only thing that's going to stick is the antigen that matches my capture, right? Then I get a primary antibody, right? That's going to look for the epitope. So a different epitope on the same antigen. And then I get my secondary antibody, right? So the rest of this is just like indirect. So sandwich has a capture and then indirect on top of it. You can have direct sandwich where the detection antibody has the color label on it as well, right? And competitive just competes for the epitope sites. So I'm not going to go much into competitive. Sandwich is what we use most. So the pros for sandwich is high specificity. Okay, it makes it a little, or not a little, a lot more accurate. All right, so let me go to the top picture again. So this is a pregnancy test. So if you think about it, it goes like this. So HCG, right here, HCG is a protein produced in the urine of pregnant women. So if we have our pregnancy test, there's a spot where you put the urine drip, right? And the urine goes, and I have these right here, right? Those right there. These are your primary antibodies, right? So it's primary antibodies. So my antigens are not attaching to a plate. They're being wicked across or moved across. And if they match, they're going to attach with my primary antibodies. So if you look at right here, I have antigens attached to my primary antibodies. But I also have these, right? These are my captures. So those are my capture antibodies, right? So my captures, so if my antigen meets my primary, it'll release it. And then it gets, on the test line, it gets caught by a capture, right? So the capture, if the capture has it, then it turns a color. So then I have my primary antibodies here. Now these captures, the second capture captures the antibody, not the antigen. So it captures antibodies that are unattached to antigens, right? So that's my control line. Right, so in a pregnancy test, you get two lines. One that says you're pregnant, and the other line says, well, yeah, the test worked. Right, so that's actually your negative. So if you have both lines, you're positive. If you only have the one line, it's negative. It just tells you that the antibodies were present and you had enough flow to get it across the control line. Right, so a lot of tests kind of work in that direction. So if you looked at this test, and this is important because I will ask it, so this pregnancy test is a direct sandwich test. So this is a direct. If it was indirect sandwich, I would need another antibody to attach to the top of this. Right, so if it was indirect, I would need another antibody to touch. But this is direct sandwich. Okay. So let me come back down. All right. So again, this is sandwich right here. So if you look at this one, right? So this example here, I've got my capture antibody, I got my antigen, my detection antibody, and then I've got another one. So this one is my secondary. So this would be so that's my indirect sandwich. Okay, and again, I have my enzyme here, my HRP enzyme, that when I give it TMB, it changes to the color blue. And we use that one quite often. All right, reading, Elisa. So a lot of reading is done just visually, but I can use a spectrophotometer, right? According to Beer's law. So remember I showed you up here, this plate, right, there are different colors based on differences in amounts of antigen present. So I can get a plate reader, and that plate reader will read the different intensities. So you'll get a spike at a certain wavelength, but the intensity of that spike will be the intensity of color. So, all right, well, hope that was slightest bit interesting, and you'll have an activity, and you'll get to investigate what we're currently using for antibody tests for COVID. All right, hope you have a good day and you're staying well, and we'll talk to you soon.